everyone, welcome to the course on medical biomaterials. Today we are going to talk about the various analytical tools that are used in area of biomaterial research, and a lot of tools are being used. So, we are going to spend quite some time in this particular topic. So, if you look at the biomaterials, it is highly interdisciplinary in nature. We have materials expertise, that means metals and ceramics and so on. So, maybe a metallurgist or a material scientist. We have a polymer expertise, that means a polymer chemist or a polymer technologist. We need experts from chemistry, organic chemists. Then we need experts from biochemistry who will be doing a lot of biochemistry assays. Then we need expertise from biology who will look at immune responses, blood responses and so on. Then we need support from engineering, who will be designing material. Then all these expertise join together, and which leads to a particular biomaterial. Okay. We also will of course need at a later point of time clinicians, medical practitioners, surgeons and so on, if you are going to take your biomaterial further down into the final market. So, because of being an interdisciplinary field, the instruments, the tools that are used for analyzing the material are wide and vast. So, it is not possible for one type of expert to comprehend all these, but we need expertise from various disciplines, as you can see from here. Okay. So, what are the tools used in biomaterial research? Okay, quite a lot. Okay, you want to see the morphological changes, more like physical changes. We want to look at whether the material has swollen, whether it is shrunk, or there is a deformation of the material because it is in contact with the body fluids, maybe for a short duration or long duration. So, we need to carry out those type of tests. Then we need to go into scanning electron microscope to look at the morphological changes in micron level. We may have to go to atomic force microscope to look at it in nano level. So, there is a lot of tools that are being used if you are looking at morphological changes. We will spend more time on the scanning electron microscope and atomic force microscope later as we go along. Then we are looking at weight loss. Suppose there is a degradation of the material or resorption, you would like to see whether the material has lost its weight. If it is a drug delivery system, you want the material to actually lose weight due to bioresorption. Okay. Then there are going to be a lot of surface changes on the material. Okay. Material may lose its hydrophobic or hydrophilic nature. The material may lose its surface energy or gain surface energy. The material may become rough because of being present inside the body fluid. If there is flow of blood or if there is a flow of urine, they may become rough. Is that going to have any effect? So, we need to study that. Next comes changes in mechanical properties. Okay. If you are using materials especially in the bone or in the knee or where load bearing, I would like to know the stress strain changes. Is there a change in their flexural strength if you are using it as a diaphragm valves and so on and so on. So, there are mechanical properties you need to understand, changes in the mechanical properties you need to understand. Then comes crystallinity. Has the material become more crystalline, or the material from crystallinity has become more amorphous? Because of the crystallinity changes, the degradation pattern may change, some of the mechanical properties may change. So, we would like to know whether there is changes in crystallinity. Sometimes when the material becomes amorphous, it may even solubilize. A polymer, crystalline polymer may be insoluble, whereas when it becomes an amorphous polymer, it may become soluble. So, we need to understand the crystallinity. Then comes thermal changes. Okay. Has the material changed its phase, okay, from one phase to another. So, there are instruments like differential scanning, calorimetry, thermal gravimetry and so on. So, we can find out the thermal changes. Of course, in return they are all connected with crystallinity. So, we can study the material, especially if they are polymeric in nature. Then molecular weight changes. If it is a polymer, I would like to know whether the has it degraded, has it become more oligomeric in nature. Okay, so we can determine viscosity average molecular weight, or number average molecular weight, or weight average molecular weight. Okay, so there are many tools like gel permeation, chromatography, mass spectrometry, which is very useful to determine the mass, okay, of the material. And suppose if macromolecules are leaching out, or if solvents are leaching out, we can find out the molecular weight using the mass spectrometer. Different types of mass spectrometry is there. We are going to spend time something like MALDI, MS, that is mass spec, LCMS, which can separate the various components using the liquid chromatograph, then the mass spec, so on actually. Then we can look at lot of changes in surface chemistry. This is a chemical approach, because as the material is inside the body, it may get say hydrolyzed, it may get reduced, it may get oxidized, it may get attacked by reactive oxygen species. So there could be a lot of functional groups, new functional groups created, old functional groups might have disappeared. Okay. So I would like to understand that there are many tools there, like infrared spectroscopy, Fourier transform infrared spectroscopy, UV visible, Raman spectroscopy, nuclear magnetic resonance spectroscopy, time of flight SIMS, okay, surface, okay, secondary ion, this is called a secondary ion mass spec, time of flight secondary ion mass spec. So if you are bombarding a surface with the electrons, the ions that are produced, they are called secondary ions, so try to characterize them. So you see lot of these tools, physical, chemical changes that are happening to your biomaterial, surface changes, bulk changes, all these. Now in addition, we need to understand the biological changes that are happening. We need to know, has the polymer or material, solid material like metals or inorganic, has acquired attached biofilm. Okay. So I need to find out how many colonies are present in the biofilm, how many live cells vis-a-vis dead cells. I need to know how much protein is attached in the biofilm, how much carbohydrate is attached. Okay. All these quantities I need to know. In addition, I can perform the Fourier transform infrared to see what are the surface functional groups that has happened there. I can use Raman spectroscopy. So if I want to study the bacterial biofilm, that means the biofilm that is attached on the surface of the material, be it a polymer, be it a metal or a ceramic, I may have to do all these various characterization. Then I need to know whether the material is cytotoxic to animal cells. Okay. So if it is going to be cytotoxic, then we do not want to use that material. So we mean to say we need to see how much live cells are present after say 24 hours, after 48 hours. Okay. What is the percentage of dead cells? Okay. I need to do that. So there are some assays for that. Then I want to know how the cells are growing, and if I am using this material as a scaffold for growing cells, are the cells settling down and then slowly differentiating, so I need to study that. Then I need to know the hemocompatibility. That means if it is a blood contacting device, is the material going to be very toxic to the blood? Will it create responses so that the blood starts clotting on the surface, which is very, very dangerous. Okay. So you see lot of biological studies I need to look at. I need to look at bacterial biofilm related. I need to look at whether the material is cytotoxic to the animal cells, whether they induce any response, and then are they problematic to the blood plasma, blood protein, and other various components of the blood. Am I going to have coagulation, or I am going to have any other problems related to the blood compatibility. So you see, we need to have facilities for carrying out physical parameter study, the chemical parameter study, and biological parameter study. So it becomes very difficult for one maybe lab to have all these facilities. So sometimes they may have to do it in collaboration with other groups. Okay. I might have covered many of them, but not all of them. Okay. Okay. Now we will look at many of them slightly more in detail, because as I said that they have a lot of importance. Okay. So we will look at it. For example, I may have a drug delivery system, I want to know how it gets swollen as a function of time because of ingress of water. So, as the water starts going inside, say the polymeric material will start swelling, so there could be hydrolysis taking place, the polymer may be degrading either as a bulk or surface erosion, as I said. So, whatever is inside will get released, okay, whatever is encapsulated. So, I want to study how the swelling takes place as a function of time. Okay. So, it may follow a graph like this. So, as a function of time it may go up and then may flatten out. So, what do? You measure the weight as a function of time, okay, and then you plot it. Now, if the polymeric material also starts degrading, so after some time the weight may start going down. So, if that happens, you can be sure that maybe the material itself is degrading. So, if the swelling rate is faster than the degradation rate, then of course, you will not see any degradation. So, you will not see decrease in weight, but if the degradation also starts taking place, then the weight may go up and start decreasing. Okay. So, with the help of a very simple set of physical data collection, we will be able to understand the mechanism of swelling of polymeric system. Okay. So, if the material is losing weight, so you may end up having a relationship like this, no, the weight loss as a function of time, it will be going down maybe as a first order or maybe as a half order and so on actually. Again, this data is very useful, especially if you are looking at drug delivery systems. Okay. Okay, then comes surface wetting properties. This is extremely important because you want hydrophilic material, hydrophobic material sometimes cause bacterial attachment, sometimes cause cytotoxicity. Hydrophilic materials are always preferred in such situations. Okay. So, the balance of hydrophilicity versus hydrophobicity is very important. Okay. So, for example, look at this, this is a water droplet on a surface. So, material of course is highly hydrophobic, that is why the material is not, the water is not spreading. So, the more hydrophilic it is, water will spread out, more hydrophobic it is, water will not spread out. Okay. So, that is a very good measure of knowing the hydrophilic nature of the material. For example, there are instruments that is called a goniometer. Okay, it measures the contact angle a water droplet makes with the surface. Okay. So, look at this. So, a water droplet is slowly placed on a surface. Okay, this is a surface, this is a needle which puts in water. Okay, the water falls in. So, then what do? Angle that is made is measured, okay, and from the angle one can tell whether the surface is hydrophilic or whether the surface is hydrophobic. Okay. Then we can also calculate surface energy, so many things. For example, you can see a water droplet spreads very well here. Okay. So, of course, the material is very hydrophilic, whereas here as you can see the water droplet is here like this. Okay. So, this is hydrophobic, relatively hydrophobic, whereas in the previous picture I showed you, this must be a extremely hydrophobic surface because water just sits on it, it is not spreading at all. Okay. So, this is relatively hydrophobic, this is very hydrophilic. So, the contact angle, as we can see, this is the angle will be very less. So, lesser the angle, we can say the material is hydrophilic, 20 degrees, 30 degrees, 40 degrees, whereas 70, 80, 90 and so on, we can call it hydrophobic surface. So, it is very important to note this. So, whenever you design a biomaterial and you prepare, you need to check the contact angle of the material, okay, and tell whether the material is hydrophilic or hydrophobic. As I said, hydrophilic material have high surface energy. So, attachment of bacteria is very poor, and also the animal cells do not have a toxicity towards hydrophilic material, whereas hydrophobic material, the attachment of bacteria also will be very high. These animal cells could be toxic to that material, okay, or the material could be toxic to the animal cells. Okay. So, if you have a hydrophobic surface, generally try to modify the surface hydrophilic. You may add functional groups like OH or NH and so on, so that material is made more hydrophobic. So, goniometer is a very powerful tool to measure the contact angle a water droplet makes on a surface of a biomaterial. Okay. Now, let us look at it. Microscopes. Microscopes are very important. We have light microscope, we have electron microscope, we have atomic force microscope. Atomic force we call it AFM. Okay. Everybody knows light microscope. This is the normally we see under light. Okay. Of course, the magnification is very limited in light microscope, maybe 100x using oil. So, if I want to see further, if I want to see biofilms, if I want to see bacteria, I may have to go for scanning electron microscope. If I want to look at nanoparticles, I may go to transmission electron microscope or even atomic force microscope. Okay. So, depending upon what you want to do, you choose different microscope. Okay. So, if you are looking at live dead cells using some fluorescent dyes, I may stop with light microscope. If I want to look at bacteria, biofilms, I may have to go to scanning electron and so on actually. Okay. So, these are the various categories of microscope: the light microscope, electron microscope and atomic force. So, I can see in nanoscale 1 nanometer with AFM. Even with TEM I can see between 1 and 5 nanometer, whereas SEM I can see 100 nanometer. Light maybe it could be millimeter going right up to micrometer. Okay. So, this is what is microscope, and microscopes are widely used in various areas of biology and especially the area of biomaterials. It is widely used. Okay. Okay. Let us look at each one of them little bit in more detail. The scanning electron microscope. What is the scanning electron microscope? So, here a stream of electrons from an electron gun or a source is accelerated. Okay. So, it is accelerated by using some voltages, and then there is a magnetic lens. So, it is focused. This is all done through a positive electric potential because the electrons are negative. So, it is focused through a magnetic lens and then hits the sample. So, secondary electrons are reflected or backscattered. Okay. It could be even photons of characteristic X-rays and light. So, here you have a detector, and from the detector you measure it. Okay. So, we have an electron source, a beam is produced, focused, it hits the sample. So, you have backscattering electron, secondary electrons, photons, X-rays, all these are produced, it is detected here. So, we can look at samples going right up to 100 or even 50 nanometer scale, not below that. So, for example, this is say a polymeric surface we are looking at approximately 100 nanometer scale. We can look at see rough surface. So, because of being present in a body fluid for a long time, it has acquired roughness. That is called a scanning electron microscope. Okay. Now, with scanning electron microscope we can also do compositional analysis. That means I can look at the elemental composition on the surface using an attachment called EDX or EDS. That is called energy dispersive X-ray spectroscopy. So, we can attach that. So, a high energy beam of charged particles, electrons or protons or X-rays is focused on the surface. Okay. So, at rest an atom within the sample contains ground state electrons in discrete energy levels or electron shells bound to the nucleus. So, incident beam may excite an electron in an inner shell, ejecting it from the shell while creating an electron hole. So, an electron from an outer higher energy shell then fills this hole, and this difference in energy between the higher energy shell and the lower energy shell is released in the form of an X-ray which is detected. That is why it is called energy dispersive X-ray spectroscopy. So, what do? You focus a beam of electrons. Okay. So, it excites the electrons, okay, at the innermost shell, ejecting it from the shell. So, it creates an electron hole. So, electron from outer, that is high energy shell, fills this hole. Now, this difference in energy is liberated, okay, in the form of X-ray. Okay. That X-ray tells you because it is a measure of the difference in energy, which is also a measure of the type of element. The number and energy of the X-rays emitted from a specimen is measured by a energy dispersive spectrometer. Okay. This is how you can measure the composition of various elements present on the surface. So, energies of the X-rays are characteristics of the difference in energy between the two shells and the atomic structure of the element present. Okay. So, the energy difference is characteristics of the shells of the atomic structure of that element. So, from the energy of X-rays that are liberated, one can tell what type of elements that are present on the surface. Okay. For example, look at this picture. Okay, this is an inorganic salt deposition on a polymer. So, we are looking at magnesium, phosphor, calcium, chlorine, potassium, aluminum, carbon and nitrogen, of course. So, this is how a EDX picture looks like. Okay, like this. This is the SEM of that, and this is an EDX. So, we are trying to see the salt, what are the elemental composition. We can even get the elemental composition of the salts that are present on the surface of this material. So, we can get both the surface morphology as well as the composition of various elements present on the surface. So, what are the components of this EDX? Excitation source, of course, electron beam or X-ray, detector, pulse processor and the analyzer. So, EDX is very important. So, generally they have an attachment with the SEM. So, we can look at one area of the biomaterial surface, either a surface modified or after being implanted, explanted. Then we want to know what are the elements that are present. We perform the EDX and we get these type of spectrum. Then from here we can tell the composition as well. So, that gives you a nice idea about the composition. So, with the respect to control, we can say what changes have happened. For example, this picture, as I said, is salt deposition on a material surface, and what are the elements that are present in the salt, like phosphorus, calcium, potassium, magnesium, chlorine, okay, and then oxygen and so on. So, it is very powerful for you. We can get a feeling of the surface morphology as well as the elemental composition. Okay. Now, let us go to transmission electron microscope. It is more powerful. We can go up to nanoscale. Okay. We can look at nanoparticles, 10 nanometers, 20 nanometers, 5 nanometers and so on. So, it is extremely very good. In a scanning electron microscope, as I showed you, the detector signals come out like this. Okay. So, you will have the electron source and the detector signal on the same side of the sample, whereas in a transmission electron microscope you can see the signal gets transmitted through the sample. So, we detect it on the other side. So, obviously, in a transmission electron microscope, if you want to perform some studies, the sample thickness should be very, very minimal, because the signals have to pass through the sample. So, here a beam of electrons is transmitted through an ultra thin specimen. So, these electrons interact with these specimens, okay, and then that gives you some signals which are detected on this side. Otherwise, it is almost, this portion is almost same as the scanning electron microscope. Okay. So, this is a picture of nanoparticles produced through a chemical synthetic procedure and detected using a transmission electron TEM, as we call it, and you can see these are all in nanoscale, maybe about 20 nanometer spherical particle. So, if you want to see so nicely, we cannot use a scanning electron microscope, we need to use a transmission electron microscope. Okay. Then comes atomic force microscope. Actually, atomic force microscope is not a really a microscope like light or SEM or TEM. Okay. It is very interesting. It measures the changes surface roughness, for example, of a cantilever that moves on the surface of the material. So, it is a more like a scanning probe microscopy. Okay. It is a probe, cantilever probe, which travels on the surface, and the changes in its height due to the roughness is measured through a detector, and that picture is shown. That is what is atomic force microscope. Okay. So, it is not like a light microscope or scanning electron or a transmission electron. So, what happens is there is a probe which touches the surface, it is a mechanical probe. So, there is a laser which is focused on it. So, as the probe moves, because of the roughness of the surface, the probe may move up and down, which is captured here in photodiode and which is detected. So, this particular cantilever moves all across the surface of the sample, and its upward and downward movement is captured, and that is a measure of the surface topography or surface changes. Okay. And this is what the picture is, and we can get it in nanoscale. For example, the surface of a polymer which is incubated for 2 days in body fluid, okay, the roughness that has happened. Okay. So, atomic force microscope we can detect in nanoscale, just like TEM, but TEM is more like a microscope, whereas atomic force microscope is not a microscope, it is more like touching the surface, and the changes in the surface roughness are captured using a laser, okay, which is shown in the form of a pictorial representation, as you can see here, and that is what it is, okay, unlike SEM or TEM. But all these tools are used. Suppose I want to see if there is a bacterial attachment on the surface which are in very small level, we can see, or if there are any nanoparticles embedded on a polymer surface, or I can see the whether the nanoparticles are agglomerated or separated from each other. So, all these can be done with atomic force microscope. Okay. Whereas transmission electron we can look at nanoparticle, nanoscale drug delivery systems. The scanning electron microscope we can look at biofilm attachment, we can look at bacteria, we can even look at the biofilm architecture. Like last class I talked about the architecture of the biofilm, are there pores in the biofilm or are they smooth? So, there we can use SEM. So, sometimes we need to combine 2-3 different techniques so that we get a very good understanding. So, one technique alone might not be enough. Okay. So, what are the differences between all these? Okay. If you look at resolution, a normal compound light microscope 500 nanometer, TEM will go up to 10 nanometer, okay. Scanning electron microscope maybe 2 nanometer, atomic force 1 nanometer, resolution is also like that, magnifying power we can go up quite a lot, depth of field TEM is moderate, scanning electron is high, atomic force is also high, depth of field for a compound microscope, because you are viewing from the top we can't go very low down. Type of objects we can see living and non-living thing with compound microscope, whereas with these microscope what happens is with TEM and scanning, because we are using electron as a probe, it should have very high vacuum. Okay. Of course, there are now biological based scanning electron microscope where the vacuum levels are not very high, but still we need to have a very high vacuum in TEM and SEM. So, generally we cannot see live, we will always see dead bacterial biofilm, whereas we can see things live in a compound microscope and also in atomic force microscope, because we are not applying vacuum in AFM unlike TEM and SEM. Preparation technique, compound microscope it is very simple, scanning electron is simple, whereas TEM and atomic force we need to have very skilled operation. Preparation thickness, okay. Compound microscope we can have very thick, TEM we can go down to very thin surfaces, other two scanning and atomic we can have reasonably thin or thick surfaces. Okay. Specimen mounting, compound microscope has to be on a glass slide, TEM maybe on thin films on copper grades, scanning can be aluminum or carbon and so on. Field of view, compound it is large, transmission is limited, scanning large, atomic force is also very limited. Source of radiation, compound microscope we are using light, TEM we are using electrons, scanning also we are using electrons, atomic force we are using force. Medium, compound microscope is air, TEM is vacuum, scanning is vacuum, atomic force air or liquid. The nature of lens, we have glass here, we have one electrostatic plus a few EM lenses, electromagnetic lenses they are called. In atomic force we have laser, diode and few emission lenses. Focusing, compound microscope, it is mechanical, TEM in the we have lens, coil, current and voltage, whereas in atomic force it is mechanical forces. Magnification, changing objectives, current in the projector lens, current, okay. So we are using a voltage and current in that. Specimen contrast due to light absorption, TEM and SEM, it is because of electron scattering, whereas atomic force it becomes atom scattering. So we see a lot of difference between these 4 type of microscopy, compound, scanning, TEM and atomic force microscopy. Okay, so each of them have their own advantages, disadvantages. We use each of them at different levels for different reasons. Okay, sometimes we combine 2 of them together so that we get quite a lot of knowledge. So as I mentioned that you can look at surface morphology, we can look at size, we can look at agglomeration, with EDX we can look at even elemental composition. That means what are the type of elements that are present on the surface of the material. So all these are morphological based studies. Okay. These are not depth based studies, that means we do not look at the changes in the bulk, but more from the surface layers. Okay. It could be few microns depth only. Okay. Thank you very much, and we will continue more in the next class.